PhD work, which focused on the effects of global change related to human activity on organic matter processes in streams, and more particularly in so as you already know, headwater streams functioning is rather particular. Because the combined effect of the light limitation by trees from the riverine forest with the fast flow greatly prevents autotrophic activity. And thus, these ecosystems will be highly dominated by heterotrophic processes of organic matter decomposition, and therefore highly dependent on anaphylaxis input from the surrounding microbes. So anaphylaxis inputs from the recurrent vegetation are various and includes coarse particulate organic matter and CQL, leaves, logs that fall into the streams, but also dissolved nutrients and organic matter that are leached out from the soil during falling. So when coarse particulate organic matter fall into the streams, it will mostly leaf litter, which represents the most abundant form of it. It will be rapidly colonized by microbial communities and especially heterotrophic microbes. So mostly composed of aquatic fungi, these communities also includes bacteria and protists that participate to the decomposition of leaf litter using a large panel of extracellular enzymes, in turn releasing nutrients and dissolved organic matter. So despite that heterotrophic microbes are commonly associated with the decomposition of the leaves in order to harvest for carbon and nutrients, these communities can also benefit from the dissolved carbon and nutrients, so the latter being less expensive in terms of energy cost than harvesting the leaves. So as I mentioned, the decomposition of the leaves involves the action of the multi-complex enzymes. This includes cellulolytic and immunolytic enzymes, such as phenol oxidase and lacase, for instance, that are involved in the decomposition of cellulose and lignin that constitute the leaf cell walls. First, it allows the microbial communities to uptake carbon, but it also allows the access to the inner part of the leaf cells, where other enzymes, such as peptidolytic and phospholytic ones, will be able to harvest for nitrogen and phosphorus. In addition to the decomposition activity of organic matter, heterotrophic microbes also participate to the leaf substrate conditioning, that is to say, the colonization of leaves by biomass, microbial biomass with high nutritional value for heterotrophic level organisms, such as macroinvertebrates. This whole process allowing the decomposition and recycling of organic matter in streams and by extent its functioning. However, as you all already know, human activity through urbanization and agriculture has greatly impaired aquatic ecosystem across the world. And one of the main pollution sources is the inputs of large amount of chemical compounds directly into the streams, among which nutrients from fertilizers and xenobiotics such as pesticides are probably the most important. The pathways of entrance of both the xenobiotics and these nutrients into stream ecosystems involve point source pollution and diffuse sources. So while a major part of these compounds will reach the stream directly through effluents such as wastewater treatment plants, for instance, another major part will be leached out from the soils by runoffs during rainfall events. Other diffuse sources include the infiltration of xenobiotics in the ground to finally reach surface water through exchange with groundwaters, and or their evaporation into the atmosphere to finally reach surface water by precipitation events. And of course, we also must take into account all the inputs from the upstream extent of the stream. So considering that the pressure posed on the rivers and streams, and therefore on freshwater resources, all countries from the European Union decided to adopt in October 2000 the Water Framework Directive with one main goal: getting Europe's water cleaner. However, and despite the effort, and despite that surface water quality has greatly improved over the last 20 years, the Water Framework Directive has still not delivered its main objectives, such as the achievement of good ecological status of all streams in Europe. And as an example, is a map representing the proportion of classified surface water bodies holding less than a good ecological status. So the redder, the less streams and rivers in good ecological status there are. There you can see that there is a lot of red on this map. And according to a recent report from European water agencies, less than 50% of the European streams are in good ecological status, and mostly because of water chemical contamination involving nutrients and xenobiotics. Moreover, it has been noted that current indicators of the Water Framework Directive to evaluate this ecological status are mostly based on the community structure of certain organisms. So this involves the atoms, macroinvertebrates, and fishes, for instance, but they do not take into account any ecosystem function. Neither they include the heterotrophic microbial compartment. So in light of these evidences, and considering that heterotrophic microbes are the basis of the trophic food web in headwater streams, we can ask ourselves the question, what are the effects of water chemical contamination on microbial organic processes in streams? So in order to answer this general question, this thesis work was divided into three main objectives. So the first one, which was to determine the sensitivity of microbial communities associated with leaf litter to chemical contamination. The second one, which was to determine the relative importance of nutrients and pesticides on microbial decomposition activity. And the third one, which was to determine the resilience ability of these communities. So in today's presentation, I'm going to present you only the first two objectives. And so let's start with the first one. With as base hypothesis that the chemical contamination of streams negatively affect microbial decomposition activity. So to answer that first objective, we performed a field experiment in the Puy-de-Dôme region involving 6 different watersheds that you can see here. And each of these watersheds differed with each other in terms of land use. And based on these land use, we established 3 watershed types: urbanized watersheds in the red, agricultural watersheds in orange, and forested watersheds or control in that experiment in green. For each of these watersheds, we selected 1 upstream less contaminated site and 1 downstream more contaminated site, so for a total of 12 sites. So the experiment was carried out by immersing fine mesh bags containing Alnus leaves directly into the water of each site. And the microbial decomposition was estimated by leaf mass loss along the incubation periods, along with other endpoints such as enzymatic activities, microbial biomass, and structure. And of course, we performed physical and chemical measurements in parallel. And the experiment was repeated 4 times, 1 per season, so spring, summer, autumn, and winter. So according to the land use, we can see that both urbanized watersheds in red and agricultural watershed in orange displayed higher phosphorus concentration here in the upper right in comparison with the forested in green, and also higher nitrogen concentration, although urbanized watershed displayed a lower nitrogen concentration in comparison with the agricultural one. The results of a multi-residue analysis allowed to highlight 144 different xenobiotics compounds into the water of all the streams, including 91 pesticides and 38 pharmaceuticals. So in that experiment, these xenobiotics were expressed as potential toxicity using the toxic unique model. And results of this potential toxicity expressed as the sum of toxic uniques show that xenobiotics toxicity was higher first at the urbanized and agricultural watershed in comparison with the forested one. And interestingly, most of this potential toxicity was the results of the pesticides, and mostly the herbicides here in blue that represented more than 80% of the pesticide of the total pesticide. So Interestingly, and opposite to what we just observed in terms of xenobiotics toxicity, so fungal biomass here, expressed as milligram of carbon, fungal carbon, displayed an opposite trend to that of xenobiotics toxicity with a lower fungal biomass at the urbanized and agricultural watershed in comparison with the forested one, which might suggest an effect on the xenobiotics toxicity and fungal biomass. So when we express this fungal biomass as a ratio upstream over downstream, so a ratio of 1 being symbolized by the dashed line, and meaning that fungal biomass upstream and downstream are similar. Here we observed that a lower fungal biomass at the downstream site of the urbanized watershed, which was much more contaminated than its upstream site, again supporting the effect, the potential effect of the higher biotics toxicity on fungal biomass. However, fungal biomass measurements were only endpoint measurements that we performed at the reachable facts. So we had only one value of fungal biomass for each season. So it's in considering that physical and chemical parameters were rather different between sites, it's hard to conclude on the direct effect of toxicity on fungal biomass, given that we don't know at which point of these fungal kinetics were located for each of these sites. In contrast with fungal biomass, microbial decomposition rates, here expressed, here corrected by temperature, didn't vary much between watershed, although significantly higher decomposition rates were observed in urbanized and agricultural watershed in comparison with the forested one. But that those higher decomposition rates combined with the lower fungal biomass raise the question of a positive effect of the chemical contamination with probably communities that were more active for decomposing leaf litter. So similarly, if we express those microbial decomposition rates as a ratio upstream over downstream, here we saw that decomposition rates were higher at the downstream site of the urbanized watershed. It was more contaminated than the upstream site. So which again supports the positive effect of contamination on microbial decomposition. So given the fact that microbial decomposition rates were not explained by fungal biomass variation and neither by enzymatic activities that I don't present here, we decided to try to look at the influence of every physical and chemical parameters on microbial decomposition rates variation. And to that purpose, we performed multivariate analysis, which was a partially square regression, from which we extracted the physical and chemical parameters that influence the most our microbial decomposition rates here, corrected by temperature. And we found, thanks to this analysis, a negative relationship between the dissolved organic carbon and microbial decomposition, which here can be explained by the regulation of immunocellular enzymes by substrate availability. As I said at the beginning, microbes, heterotrophic microbes, can benefit either from the dissolved form or from carbon from the leaves. So probably, when there are a lot of carbon in the river, they tend to use the dissolved organic form that are less expensive. We also found a positive relationship between nutrients, and especially nitrogen, and microbial decomposition. And such a relationship is commonly found in literature. And such a correlation, such a positive relationship can be probably explained by the eco-enzymatic stoichiometry hypothesis formulated by Sitsabo. That is to say that because there are a lot of nitrogen in the water, well, the microbial communities tend to harvest more carbon in order to maintain a stable stoichiometry within the community. And finally, we found a positive relationship between pesticides toxicity and microbial decomposition rates, which again highlights the fact that communities were probably more efficient in contaminated sites for decomposing leaf litter. And this can probably be explained by community adaptations, such as community-induced tolerance. So, in conclusion, for this first chapter, for this first objective, we found that high nutrients and pesticides concentration that observed at the urbanized and agricultural watersheds were associated with an increase in microbial decomposition activity. However, we also found a negative relationship between the decomposition, microbial decomposition in carbon that can be explained by regulation by substrate availability. However, these variation in terms of microbial decomposition could not have been linked with any variation in terms of enzymatic activities. And however, we found we found a lower fungal biomass at the more contaminated site, but that can hardly be explained by xenobiotics toxicity because it would need full kinetics of fungal biomass. So, in contrast with our first hypothesis, we found that chemical contamination of streams positively affected microbial decomposition activity. And one explanation to that result is that communities were probably more efficient at harvesting the leaves in the most contaminated sites, probably because of long-term community adaptation, following functional redundancy, so probably resource partitioning, for instance. However, since both nutrients and pesticides co-occurred at the most contaminated sites, another hypothesis could be that nutrients compensated the effect of xenobiotics at the most contaminated sites. And this hypothesis would suppose a greater effect of nutrients over pesticides on microbial decomposition. So, that thesis, we decided to follow that second hypothesis, which brings us to the second objective of that thesis. That is to say, to determine the relative importance of nutrients and pesticides on microbial decomposition, with as base hypothesis that nutrients have a greater effect than pesticides on microbial communities and on microbial decomposition activity. So to answer that second objective, we performed a microcosm experiment here at the Université of Lyon. So 24 artificial stream channels were set up, as you can see here in the picture, and divided into two nutritive conditions, a mesotrophic one and an eutrophic one, that differed with each other by a factor of 10 in terms of nitrogen and phosphorus. The carbon concentration were exactly the same. So these nutrient concentration were established on the basis of the paper of Dodds et al. And on previously observed nutrient concentration in the field experiment. For each of these eutrophic of these nutrient conditions, we established control channels exposed to fungicide, the tebuconazole in red, which was selected because of its potential direct effect on aquatic fungi. Channels exposed to the herbicide S-metolachlor here in green, which was selected because of its high detection frequency in streams in France. And a mix of both the fungicide and the herbicide. So leaf containing bags were displayed in each channels, and prior to the experiment, all channels were inoculated by a microbial inoculum coming from an upstream forested stream from the previous field experiment. And as previously, leaf microbial decomposition was estimated as leaf mass loss all along the experiment, along with other endpoints such as enzymatic activity measurements, microbial biomass, and structure. And so the experiment lasted for 40 days, and we performed water renewals every week. So let's begin with the first results of, so you don't see well, but of the microbial decomposition rate for each of these conditions. And here we clearly observed higher decomposition rates, microbial decomposition rates in the eutrophic condition, so as a dashed box, in comparison with the mesotrophic one, those are the white box. And such differences in terms of microbial decomposition rates can be explained, in fact, by fungal biomass colonizing the leaves. So here you have the graph of the fungal biomass over time. And you can see that fungal biomass is higher in the eutrophic condition, as solid line in comparison with the mesotrophic one, as the dashed one. However, we still observe that microbial decomposition rates were higher in the control channels in black in comparison with the treatment, to only insignificant in eutrophic condition. And such differences in terms of microbial decomposition rates were not explained by differences in fungal biomass. So if we zoom on fungal biomass, but just in eutrophic condition this time, we can see that although there are no differences in terms of maximal fungal biomass at the end of the experiment, we can see that the control channel in black tend to increase slowly in comparison with the treatment. And this is confirmed here by the table where growth rates were calculated with a significantly lower growth rate in the channels, in the control channels here, in comparison with the treated one, which suggests that stress by pesticides alone or in nature tend to increase fungal growth rate, but not the fungal biomass maximum. And probably such differences might explain the higher microbial decomposition rates that we just observed previously, possibly highlighting different growing strategies and possibly that microbial communities in the control channels were more efficient at harvesting the leaves. Another explanation for that differences in terms of microbial biomass can come from the structure. So here is the non-metric dimensional scaling performed on the fungal community structure obtained by DDGR, and at the end of the experiment, they form T. And here we can clearly see significant differences between treatment and control here in black for the eutrophic condition, which highlight the relationship between microbial community structure and microbial decomposition. And interestingly, we did not observe any variation in terms of microbial fungal community structure in the mesotrophic channels, which can explain the absence of microbial decomposition. And we did exactly the same thing for the bacterial communities, and we observed exactly the same results, highlighting the relationship between microbial community structure and microbial decomposition. Then we try to look for an implication of extracellular enzymes in the variation of microbial decomposition. And so here are the results of specific phenol oxidase activity. So phenol oxidase activity corrected by microbial biomass in carbon, expressed in milligram of carbon. And again, here we see that specific phenol oxidase activity tend to be higher, significantly higher in the mesotrophic channels as dashed box in comparison with the mesotrophic one here, which again tend to highlight that communities exposed to stress are more active, at least in terms of enzymatic activity. And we also observed that phenol oxidase was higher in the channels exposed to the fungicide or channels exposed to the mix of the fungicide and the herbicide, which tend to highlight the stimulation of that enzyme by the fungicide. If we now look at the specific extracellular lacase activity, we obtain exactly the same results, except that the stimulation was indeed observed in presence of in channels exposed to the tebuconazole, but was no more observed in the presence of the mixture of both the herbicide and the fungicide, which might highlight antagonistic type interaction between the tebuconazole and the resmetiolachlor on that enzyme. So in a previous paper in which we tested the grain concentration of tebuconazole on a single fungal strain, so Alnus acuminata, and in which we measured, so this is the lacase activity again. We also observed a stimulation in terms of lacase activity exposed to growing concentration of tebuconazole, and we explained that such stimulation by a stress response mechanism. In literature, stress response mechanisms have already been shown and involve either an oxidative stress response, a general response, or detoxification mechanisms involving the decomposition of pesticides. And here we hypothesize that such stimulation that we observed not on that graph but on the previous one was probably the results of pesticides degradation. Here is the reaction catalyzed by the phenol oxidase, so that phenol oxidase and lacase also that lead to the opening of the monophenol rings. And considering that the result also possesses an aromatic ring, vasochoric ring, we might expect phenol oxidase and lacase to degrade the tibuconazole by that ring. So in conclusion of that second objective, so we found no effect of the pesticide in the mesotrophic conditions. And we also observed that nutrients were the main parameters positively influencing microbial decomposition. That can be explained by an increase in fungal biomass, changes in community structure, but also by an increase in terms of extracellular enzymes. Interestingly, we found an effect of the pesticides, but only in the presence of high nutrient concentration, which that time was characterized by a negative effect on microbial decomposition, along with changes in terms of community structure. And interestingly, we also observed changes, variation in terms of extracellular enzymatic activities, but that were not linked with any changes in terms of microbial decomposition. And so we explained such variation by probably stress response involving detoxification mechanisms. So, as stated as first hypothesis, we indeed found that nutrients have a greater effect than pesticides and microbial communities. However, the fact that pesticide effect was only observed in heterotrophic condition is quite intriguing. And one explanation for that result could be that the effect of pesticides was overwhelmed by the low nutrient concentration that we set up in the mesotrophic channels. And that hypothesis can be supported by the every week nutrient dissipation, because every week we changed water and we measured nutrients, and every week nutrient dissipation was at 100%. So maybe that there were lack in nutrients from microbial communities that led to a stress that was higher than that of pesticide. Another hypothesis is that nutrients can exacerbate the effect of pesticides, which would highlight that the composition of nutrients that we just talked about is only partial. And that hypothesis can be supported by the work of Fernandez et al., in which they assess the effect of a range of zinc concentration and phosphorus concentration on microbial decomposition. And they observed that the strongest inhibition was in the presence of the highest zinc and the highest phosphorus concentration. And they explained that inhibition by the fact that microbial species that were the more stimulated by nutrients were at the same time the more sensitive to the pesticides, to the zinc in their experiment. So now let's move on to the conclusion of that thesis work. So we found in that thesis work that nutrients were the main parameters positively influencing microbial decomposition. That can be explained by an increase in fungal biomass along with changes in microbial structure and an increase in terms of lacase activity. However, we did find an effect of the pesticides in with nutrients that was characterized by a negative effect on microbial decomposition that can be explained by changes in community structure. And that raises the question of the exacerbation of the effect of pesticides by nutrients. However, the fact that we did not observe such inhibition in the field can be explained by the fact that probably communities either that because nutrients compensated the effect of pesticides, or because communities in the field have experienced long-term adaptations along with more efficient resource partitioning, leading to more efficiency in terms of microbial decomposition. And we also found that microbial communities were able to display a stress response to toxicants, characterized by an increase in lacase activity, possibly involved in the detoxification mechanism. So now let's move on to the perspective. So as we saw previously, uh, uh, the interaction between nutrients and pesticides is not quite clear yet. While most of the literature found that nutrients usually compensate the effect of pesticides, we also showed that there can be a kind of exacerbation of pesticide by nutrients, and it's not quite clear what are the phenomena that trigger either of these responses. So that can be either a threshold, that means maybe higher nutrient concentration or lower. Well, maybe it's threshold dependent, or maybe it's because of the community structure as shown in the work of Florent et al. So in order to test that hypothesis, it would be interesting to test the effect of a gradient of nutrient concentration in the presence of pesticides on two types of communities, and that in such experience could confirm the or not the microbial structure related concentration of nutrients over xenobiotics. Uh, Another thing is on the field, we assessed the potential toxicity of xenobiotics using the toxic unique model, which is a concentration addition model. And despite that, it is known as a very conservative model and as one of the best to describe very complex mixture of xenobiotics. It suffers a number of limits, and one of them is that there are almost no data available. One of them is that it is based on the EC50 for the compounds on the organism. And because there are no data available on fungi, we used in that experiment algae. And considering that more than 80% of the pesticides were herbicides, we may have overestimated the effect of that in our study. And so that raises the question of setting up the hyphen effect as organisms as biological. The other problem is that while that model is used for generally one organism, uh, and considering that at the community level we have huge differences in comparison with at the species level, so the question is that model accurate enough at the community level? And so maybe it would be more interesting to determine directly on the considered community for at least the main compound detected, rather than relying on one single. In that thesis work, we found a stimulation in terms of lacase and phenolics activity to tebuconazole, and we hypothesized that such stimulation could be the result of stress response set up by fungi, and especially detoxification mechanisms, probably leading to the decomposition of pesticides. However, in that experiment, we did not measure for tebuconazole metabolites, so it's quite impossible for us to state on that specific response. And considering that there are many other that lacase and phenol oxidase are implicated in many other kind of responses, it would be interesting to directly test the detoxification response, and so the involvement of these lacase in tebuconazole decomposition, degradation, by testing directly the ability of fungal communities to degrade the tebuconazole following the metabolites, and also the enzymatic activities that could give us that could confirm or not the involvement of fungal enzymes in the detoxification mechanisms. So another thing is the title of that thesis is about the global change, but in fact we only assess the effect of water chemical contamination, but there are many other aspects of the global change, and so it's only the freshwater pollution, and the probably most known of them is the climate change, which and forecasters predict less precipitation the first 50 years, along with an increase in water temperature by 2 degrees. And this might have an effect on the drought event, so increasing the drought event in streams. And while some papers already dealt with the interaction of drought events and water chemical contamination, such as the fungicide, it was in Pest et al. None of them tested the triple interaction between nutrients, pesticides, and drought events. And in fact, this could be interesting to test that triple interaction, because it will give a better insight on the effect of global change on the ability of microbial communities to decompose organic matter. So in that thesis, we focused mostly on the microbial aspect, microbial decomposition. But the question that can be asked is, what are the effects on the higher trophic level organisms, such as macroinvertebrates, that rely on microbial colonization of leaves? So in the second objective, in fact, we kind of tested the effect of the indirect effect of pollutants on higher trophic level organisms, because at the end of the experiment, we recovered leaf discs so that were exposed to both the community colonized by the communities and exposed to the fungicide, and we presented them to gammarids, and we measured their palatability for gammarids. And what we obtained is no differences in terms of gammarus feeding rates, either in terms of nutrient conditions or between treatments. And such absence of differences in terms of gammarus feeding rates can be explained by the low exposure time, because it was only a one-day experiment. However, if we now try to correct this by expressing as a consumption, as a microbial carbon consumption per gammarus, we observe that gammarus tend to ingest more fungal carbon in the mesotrophic condition, in the eutrophic condition, sorry. And we also observe that the mix tended to be lower than the control, which probably highlights an effect of the mixture on the palatability of leaves for macroinvertebrates. But so, as I said, the problem is that we were not able to highlight any significant effect of such pesticides adsorbed on leaves on the feeding activity of gammarus. That can be explained by the short time of exposure, along with the lower adsorbed pesticides on the leaves. But it could be interesting to test the effect of longer exposure to contaminated leaves on macroinvertebrates, to see the long-term effect of food on those organisms. So to conclude on that presentation, just a small slide about me. So that thesis work is described in microbial ecotoxicology, in which I was brought to use different techniques, such as in situ laboratory microscopes, and developing many skills: molecular biology, microbiology, and chemistry. This thesis led to 6 peer-reviewed publications, along with 5 oral communications in international and national congress, and 1 poster, and that available for a postdoc position. So thank you for your attention.